Welcome to this video on the amino acids, the alphabet of life. Proteins, as you know, are essential to life and their structure and functions are encoded using a unique biological alphabet system known as amino acids. In this video, we will explore the general structure, nomenclature, classification, stereoisomerism, and functions of these amino acids. So regarding the basic structure of amino acid, an amino acid has alpha carbon atom attached to it is an alpha amino group, a carboxylic group, a hydrogen atom and a unique R group. There are 22 different types of R group, therefore there are 22 different types of amino acids. These amino acids are used in the synthesis of protein, therefore they are also called as proteinogenic amino acids. The main source of these amino acids is our dietary sources. At physiological pH or physiological conditions, amino and carboxylic groups are ionized, forming a neutral molecule known as deuterion. Only L-stereoisomers are active of amino acids. We will explain what is the basis of L-stereoisomers or stereoisomerism in the next slide. Stereoisomerism or stereoisomers are the product of the chiral carbon atom. A carbon atom which is attached to four different groups is known as chiral carbon atom or asymmetrical carbon atom. These such carbon atom will form the isomers. Two type of isomers we will explain in the next slide. And the carbon which is not attached to four different groups is known as symmetrical carbon atom or achiral carbon atom. Such molecule will not form stereoisomers. So stereoisomers is the property of a chiral or symmetrical carbon atom in organic molecules such as amino acid and also carbohydrate. So L-stereoisomers of amino acids are functional. They are formed they are defined by the position of amino group on the chiral carbon atom. In amino acid, you have one carbon atom which is chiral carbon atom or asymmetrical carbon atom. When amino group is present on the left hand side, this is known as L-stereoisomers, which is the functional which is present in our body. The other form is D-stereoisomers, which is defined by the position of amino group on the right hand side. These are not active in our system. Most of the amino acids exist as L-stereoisomers. They are functionally important in this configuration as well. Why the system has evolved or chosen L-stereoisomers, this is all hypothetical answer is there, and this may be because the enzymes or proteins which utilize amino acid have been optimized to use only L-stereoisomers during the course of evolution. This is hypothetical answer. Except glycine, all other amino acids are able to form stereoisomers. Glycine cannot form stereoisomers because there is no chiral carbon atom in this molecule. All other amino acids, as I said earlier, have chiral carbon atom, therefore they are able to form stereoisomers. So amino acids are connected by a covalent bond known as peptide bond, forming a polymer of protein. The bond is formed between the carboxyl group of one amino acid and amino group of the another amino acid, forming an amide or covalent bond which is known as peptide bond. This reaction is a condensation reaction which is associated with the liberation of water molecule. So peptide bond is a covalent bond, as you have seen in single covalent bond, but because of the electronegative atoms in this bond, There is a dislocation of electron cloud leading to the appearance of the double bond character. Therefore, the peptide bond is rigid, planar, and have double bond character. The plane is consisting of 6 atoms, and this plane has this plane can make two angles. They are known as phi angle and psi angle. These angles are also called as dihedral angle. The plane makes angle with respect to the flanking alpha carbon atom in the molecule. So we have seen that there are 22 different type of R group. Therefore, there are 22 different type of amino acids, including pyrrolysine and selenocysteine. So basically, you can say that R group defines the property of amino acids. In this case, these two amino acids, pyrrolysine and selenocysteine, they are not present in each and every protein. They are rarely present. Therefore, in textbook you will see only 20 amino acids, not 22 amino acids. So these 20 or 22 amino acids, these are proteinogenic amino acids. They are used in the synthesis of protein in our cell. So amino acids perform a variety of very important functions, including their major role in translation of protein synthesis, in the in the in supplying energy to a body. Although they are not the major source of energy or preferred source of energy, about 10 to 15% of daily energy needs is supplied by these amino acids. They also act as some of the amino acids such as glutamate act as neurotransmitter. It is excitatory neurotransmitters, whereas you will see some act as precursor of neurotransmitters such as serotonin, melatonin, dopamine, which is required for our mood concentration and also for the regulation of the sympathetic and parasympathetic nervous system by epinephrine and norepinephrine. These the precursors for these neurotransmitters are mainly aromatic amino acids. They also act as the, the precursor for the synthesis of melanin pigment for our skin, eyes, and hair. And also they also act as precursors for purine pyrimidine synthesis, which are required for DNA RNA synthesis, and also for the histamine. This is biogenic amino acid, biogenic amine, sorry, which is a very important molecule for the allergy or hypersensitivity reaction. In addition, thyroid hormone or T3 is also synthesized utilizing amino acid. And in the end, this is also the certain amino acids are play a very important role in detoxification of ammonia. These are glutamate, glutamine, and alanine. These three amino acids play very important role in detoxification of ammonia through urea cycle. So this is all about the functions of amino acids. So regarding the nomenclature of amino acids, the name of amino acid which we have heard in biochemistry, they are traditional, or these are classical names. Nomenclature here, I mean how you represent amino acid in a short hand notation. There are two ways amino acids are represented in shorthand. First is using three-letter code word, and second method is using one-letter code word. So regarding the representation of amino acid using three-letter code word, I would like to emphasize here that in most of the cases, the, the first three letters have been used to denote an amino acid. However, there is discrepancy which I would like to explain here. The first group, which start with A, is aspartic acid and aspargine. In both cases, the first three letters are same, therefore you cannot assign ASP for both. ASP has been chosen for aspartic acid, whereas aspargine has been given or assigned ASN. The next group is the glutamic acid and glutamine. The glutamic acid and glutamine both have same first three letters. Therefore, you cannot assign GLU for both. So GLU has been assigned to glutamic acid, whereas GLN is for glutamine. The last pair is tyrosine and tryptophan. In these two cases, although the first three letters are different, but TYR and TRY, they are very similar and confusing. Therefore, to avoid confusion, tyrosine has been given TRP, and tryptophan, sorry, has been assigned as TRP, whereas tyrosine has been assigned TYR. So these are three cases where you see the three-letter code is not representing the first three letters. The first one is asparagine ASN, glutamine GLN, and tryptophan TRP. So second method of representing these amino acids is by using one letter code word, and in most of the cases the first letter which is used. However, there is some discrepancy which I would like to highlight here. Now I have arranged these amino acids according to alphabetic order. So the first four amino acids, they start with A. So alanine has been assigned as A, whereas the other amino acid, the remaining three which start with A, have been assigned different letters. For aspartic acid it is D, for asparagine it is N, for arginine it is R. And the second group start with G, so glycine is G, glutamic acid is E, and glutamine is Q. So these are again the discrepancies. And the third pair is leucine and lysine. Leucine is represented by L, first letter, and lysine is represented by K. K is close to L, therefore K has been chosen for lysine. The next pair is proline and phenylalanine. Proline is P, phenylalanine is F. And the last group is which start with T. So threonine is T, tyrosine is Y, and tryptophan is W. So this is nomenclature using one letter code word. One letter code word and three letter code word, they are commonly used to write or represent amino acid sequence of a peptide or protein. So they are very important to know. So coming to the classification of amino acids, they have been classified based on three criteria. The first one is based on R group properties, second is nutritional requirement, and the third criteria is catabolic or metabolic fate of the amino acid. So the first classification is based on the properties of R group. This is basically based on the interaction of these amino acids with water and other molecules. These are five different categories in this. The first one is acidic amino acid. There are two amino acids. The side chain is acidic. The second is aromatic amino acids. These are three amino acids. The side chain is aromatic in nature. The third category is basic. There are three amino acids having basic side chain or or positively charged side chain. The next is non-polar. Here I would like to emphasize that non-polar has nothing to do with the charge. It simply means that the side chain is aliphatic and hydrophobic in nature. There are 7 amino acids in total in this category. The fifth one is polar. Again, polar doesn't have anything to do with the charge. It simply means that these are hydrophilic. They have hydrophilic side chain. There are 5 in total. So these are 20 amino acids divided into 5 different categories based on their interaction with water and other molecules, which is important for our understanding of protein folding, enzyme activity, and molecular interactions. The second classification is based on the nutritional requirement, means whether our body can synthesize these or they are required through dietary sources. So there are two type of amino acid or two groups of amino acid. First one is essential amino acid, second is non-essential amino acids. So the first category is essential amino acids, means our body is unable to synthesize these amino acids, therefore they must be supplemented in our diet. So these are 9 amino acids and you have to memorize it. I have defined or designed a mnemonic to enhance your memory. Try to move to the hill top fast and vigorously. These letters, which are red in color, representing amino acids. So you can write down methionine, tryptophan, threonine, histidine, isoleucine, leucine, lysine, phenylalanine, and valine. So these are 9 essential amino acids. They must be supplied in our diet. If they are lacking in our diet, we will develop the symptoms of deficiency or diseases. The second category is non-essential amino acids, means body is able to synthesize them and they are not essential to include in our diet. These are 11 amino acids which are listed here alphabetically. All 4 amino acids with A first letter: alanine, arginine, asparagine, aspartate, cysteine, and all 3 with G: glutamate, glutamine, glycine, proline, serine, and tyrosine. So these are 11 amino acids which are non-essential. Now the last category is of the classification is based on the catabolic fate. When we talk about catabolic fate or metabolic fate, means they are going to supply energy, right? As I mentioned, that they are not major source of energy, they are not preferred source of energy, but they do supply about 10 to 15% of daily requirement in our system. In addition, their role becomes significant in prolonged starvation and in strenuous exercise. So there are basically 3 different categories in this class of classification. The first category is ketogenic amino acids, including 2 amino acids only. There are 13 glucogenic amino acids and there are 5 which have both properties as ketogenic and glucogenic. So the first category is ketogenic amino acid, which means that these amino acids are metabolized finally to produce ketone bodies, which are converted to acetyl-CoA and enter Krebs cycles to produce energy. So these are 2 amino acids, leucine and lysine, which are strictly ketogenic, and it is easy to memorize and remember these amino acids. So the second category includes glucogenic amino acids. There are 13 strictly glucogenic amino acids. These amino acids are metabolized finally to the two different intermediate of Krebs cycles, which then enters the Krebs cycles to provide precursors for the synthesis of glucose by gluconeogenesis, and also they supply energy in the form of ATP. So basically these are 13 and it is difficult to memorize, but I will give you an easy way to circumvent this problem. So last category has a group of 5 amino acids which has properties of being glucogenic as well as ketogenic. These are tyrosine, isoleucine, phenylalanine, threonine, and tryptophan. They are basically they are either metabolized to ketone bodies and they enter Krebs cycle as acetyl CoA, or they can also enter Krebs cycle through formation of various intermediate of Krebs cycle, and therefore they provide precursors for the gluconeogenesis. So these are 5 amino acids which have both properties of being glucogenic and ketogenic amino acid. You can memorize these 5 amino acids using this mnemonic. Tyrosine is fierce, tough, and terrific. Each highlighted letter is an amino acid. Tyrosine isoleucine, phenylalanine, threonine, and tryptophan. So this is all about the classification of the amino acids. So in summary, amino acids have been classified based on three criteria. The first one is based on our group properties into five different groups: acidic, basic, aromatic, polar, and non-polar. This is based on particularly the nature of side chain and also their interaction with water molecules. The second category includes all the criteria is based on nutritional requirement. They have been divided into two categories: essential amino acid and non-essential amino acids. There are 9 amino acids which are essential. Non-essential amino acids include 11 amino acids. The third category is based on metabolic fate. There are 2 amino acids which are strictly ketogenic. There are 13 amino acids which are strictly glucogenic amino acids, and there are 5 amino acids which have properties of being both ketogenic and glucogenic. So finally, I would like to emphasize that this is a high-yield topic. You will find questions in all examination, including entrance exam or end of term or end of year exam, and this will be helpful. Therefore, I request you to like it, subscribe it, and share with your friends who are interested to learn amino acid and protein structure and function. So thank you once again. Until next time, take care. Bye bye.